Welcome students. I hope today we are going to continue with that various chromatographic techniques which have been used or in herbal standardization and its application. We have seen HPLC, TLC and even ultra HPLC, and also we have seen hydrophilic interaction, liquid chromatography, and today we are just going to see some of the important sessions in chromatography like gas chromatography, and even the old one, the column chromatography. Have to also, I did not give some applications. Sometimes if they ask short notes, the student have to write that. So I have taken that also as a consideration. And I hope my students, I have been your mentor, your teacher and a second family, and best for your bright future, because I hope when this is our mostly after 2-3 sessions we will wind up our classes, I think so, because only one chapter is left, that too I think 1 or 2 chapters with some datas of applications and about some pharmacovigilance also, how, and also some pharmacopoeias we have to have a discussion. I'm not winding up today, but I will just continue. But still I remembered about how I spend my time with my students. So I want to just give you for a bright future, trying to explain how much I can. So today what we are going to see is about a short application of gas chromatography and a column chromatography, slightly hidden, and supercritical fluid chromatography, SFC, and two-dimensional chromatography. What is two-dimensional chromatography? And various hyphenated detection techniques. And hyphenated techniques are nowadays more common, and it has more application in various sectors, not only in herbal drug technology and herbal formulation or herbal identification of the drugs, qualitative and quantitative formulations and herbs. But apart from that, there are various other technology also. We are depending on hyphenated detection. Hyphenated detection means combining 1-2 instruments. In online means, we are using this word online means you should not interpret with the internet. Online, that means one compound which is coming from one chromatography or one instrument, it will be taken to the another technology, hi-fi technology can be connected, like GCMS and LC-NMR and gas chromatography with NMR, and liquid chromatography with NMR, liquid chromatography with mass spectroscopy. Like that we are connecting. This is what today we are going to see about these hyphenated detected techniques. They may ask you sometimes the questions like that. Can you explain about hyphenated detection techniques which you have studied? So in that case, you should be able to write about the GCMS or LC-NMR. Even why not? If it is even IR spectroscopy is also even connected with liquid chromatographies, gas chromatographies. But it is sophisticated technique which all many type of researchers and many type of scholars are working in this instrument analysis to get a very, very fast analysis result in herbal medicines. You know that more than 80% of the population in the world now are depending on herbal drug treatments. And herbal drug, they are using the advantages of herbs in various sectors. So now we will have a discussion about the small discussion about gas chromatography, and it is actually GCMS, gas chromatography with mass spectroscopy. This is also hyphenated, an online system which is from gas chromatography and mass spectroscopy are connected. Anyhow, gas chromatography is alone is also we have application. So we just made it. For example, this picture shows a gas chromatography with mass spectroscopy, a hyphenated detection of two instruments. So you can see the picture that it says inlet of helium or a mobile solvent. Mobile phase is a gas, and which is also having the same operates in the partition chromatography, because you know very well gas and liquid are having the same physicochemical properties almost when they are applied in various sectors. In physics you might have studied about that. So the inlet of a mobile gas can be given in the picture which we have shown there, and an injection part, same like you have to know about the septum or rhodamine or injection part where the sample is injected and it enters into the column. It is bound a long column of a thin column, long column of, maybe you might have studied about gas chromatography, solid chromatography, that means GSC and GLC you might have studied when you are studying in the chromatographic technique in chemistry. So GLC, which is solid, which can operate in an adsorption chromatography, and GLC is gas liquid chromatography, GSC is gas solid chromatography. So according to the packing material in the column, whether it is a solid or it is a solid impregnated with a liquid which acts as a stationary phase, so that makes the principle behind the column chromatography and the gas chromatography. And this gas chromatography is a hyphenated program or an online system. A two gas chromatography is connected with mass spectroscopy as shown in the picture. So once the compound is eluted, compound is separated from the column of the gas chromatography and enters into the ionizing source, that ion source, which is, you can see there is a coil which is having there, by means of heating technology with the coils, and ionized, the compound is be ionized or combusted, and then the ions are mass filtered and can be detected, and so that you can identify the mass number and the mass of the compound which has been eluted from the chromatogram. The fingerprint is very interesting, but the chromatogram is very interesting to get from GCMS, and it is a very sophisticated instrument for more than 3 decades. Starting from chromatography, we can see this gas chromatography connected with mass spectroscopy is one of the instruments which was given lots of papers, and lots of researches have been done by GCMS. So anyhow, we are giving a short application with just 2 slides. I am just explaining about gas chromatography. Gas chromatography, you can see, is a well established analytical technique commonly used for characterization, quantization, that is quantification or quantization amount, and the identification of volatile compounds. So when you are going for volatile compound identification in other spectroscopy, other chromatographic techniques, it having lot of disadvantage, but here only volatile substances will be very fast and very feasible separation, and also very good results can be obtained in the gas chromatography, as the gas is a mobile phase. It can be used for different fields such as pharmaceutical, cosmetics, and even environmental toxics study can also be done by gas chromatography. Since the sample have to be volatile, human breath, blood, saliva, human breath, blood, saliva can analyzed by means of gas chromatography, and other secretions from our body containing large amount of organic volatiles can be easily analyzed by gas chromatography. So it has a lot of application in laboratory of gas chromatography. And you can see the powerful separation, efficiency and selective detection makes gas chromatography a useful tool for the analysis of essential oils. Despite its advantage, gas chromatography analysis of herbal products is usually limited to the essential oil because of possible degradation of thermolabile compounds. So thermolabile compounds may be because of temperature and because of the flow property of the gas which is moving through the column. The thermolabile compounds may be degraded. There is some limits of gas chromatography application. The requirement of volatile components make gas chromatography unsuitable for many herbal compounds. So every components or every instruments there will be an advantages as well as there will be a disadvantages. So there are some limitations, that's what we have mentioned here. And when we talk about gas chromatography connected with mass spectroscopy, that is hyphenation detection in online, from gas chromatography to mass spectroscopy. Online means, I don't mean that is internet. Online, from the compound which is separated from gas chromatography enters directly in an on-line, not online together. Leads to reducing analysis time. Analysis time is reduced in essential oil 40 to 100 seconds is enough, as well as decrease the detection limits. Gas chromatography with mass spectroscopy, that is hyphenated analysis of essential oil, showing faster analysis and high efficacy or high efficiency. Made use of microbore capillary columns with reduced stationary phase, film thickness, very thickness, mild thickness, very thin, and 10 meter long of the column with 100 micron and a 5 meter into, and also you can have 5 meter long, 10 meter long can be used with, if a 10 meter long with 100 micrometer internal diameter can be used, and 5 meter long and 50 micrometer internal diameter can also be used. With a rapid temperature program of 20 degrees centigrade per second, fast data, acquisition of using the flame ionizing detector, which has been used to get then ionizing, then immediately it is connected with, by means of mass spectroscopy in the online, a high split ratio can be obtained. Finally, the low pressure GCMS gas chromatography connected with mass spectroscopy by means of hyphenation, that is our hyphenation detection technique by means of two instruments in online, using megapore analytical column of 10 meter long of 530 micrometer with 0.25 to 1 ml of film thickness can be also used for megabore analytical techniques. Was investigated on the essential oil and leads to a slightly reduced efficiency but manifold decrease analysis time. The main thing is the analysis time is more reduced, and high efficiency or high efficiency results can be obtained by means of GCMS, gas chromatography mass spectroscopy. And gas can be used to determine the identity of natural products containing complex mixtures of similar compounds. For example, the geographic source of a crude oil or a natural gas can be determined by the fingerprint, by the chromatograph, or the relative distribution of major and the trace compound in each oil can be determined. Naturally produced oil such as food products and fragrance can be identified by GCFID and GCMS. That is, flame ionizing detector connected with gas chromatography or gas chromatography directly connected with mass spectroscopy can also be used in hyphenated technique, hyphenation technique. So that is what you can see. There is gas GCFID means gas chromatography connected with flame ionizing detector, or you can have a hyphenated techniques of online by means of connecting two instruments like gas chromatography and mass spectroscopy. For an example, we have given an example. For example, the sample of 0.25 cc natural gas sample is injected, and we can find the separations and identification of the compound using a carrier gas. The mobile phase is helium, 8.6 ml per minute in 60 degree centigrade. The column which is used to be HP plot Q, 30 meter long into 0.5 millimeter and 40 micrometer internal diameter. Very, very tiny. And the injection split mode should be 100 ml per minute, and detector which you are using for this purpose is TCD, that is thermal conductivity detector, can be used. This is an example for natural source. Just you can mention for gas chromatography with mass spectroscopy or gas chromatography with a detector of thermal conductivity detector. Just a gas chromatography example. And now we want to discuss about hyphenated detecting technique, not only by gas chromatography, mass spectroscopy. There are many other techniques are also there. So there may be asked about what is hyphenated detection techniques and what all the things which can be followed. If they ask, the student have to be able to write this one. That's why I took this one as a small part of 2 slides to explain about hyphenated detection techniques. Just now we discussed about mass spectroscopy, which is connected with gas chromatography. And you can see hyphenated technique means is developed from the coupling or 2 or even 3, 2 usually, coupling of separation technique and an online. That means online means we are telling about from one instrument to another instrument is separated from the gas chromatography directly the compound enters into the mass spectroscopy, or the liquid chromatography directly enters into the mass spectroscopy, or a liquid chromatography connected with NMR directly connected, that is also possible, or a liquid chromatography directly connected to the IR spectroscopy is also possible. So that's why we are using this online spectroscopy detection technology. The remarkable improvements in the hyphenated, you have to remember about this word, hyphenated analytical methods over the last 2 decades have significantly broadened their applications. In the analysis of biomaterials, especially natural products, in this article, that means especially I have taken from an article, they have mentioned recent advance in the application of various hyphenated techniques like gas chromatography connected with mass spectroscopy, liquid chromatography connected with mass spectroscopy, LCMS, GCMS, LCFTIR, liquid chromatography connected with Fourier, infrared, that is Fourier transformer of infrared spectroscopy, liquid chromatography connected with NMR, nuclear magnetic resonance, and a CE connected with MS. Capillary electrophoresis is connected with mass spectroscopy. Capillary electrophoresis separation of technique which is used for high electric field to produce electro-osmotic flow for separation of ions. So CE is also connected with mass spectroscopy. What do you mean by CE? Sometimes if they ask, you should know that capillary electrophoresis, which separation technique, it is also a technique of chromatographic techniques for separation, but electrophoresis by means of charged atoms and charged ions of cation and anion, which use for high electric field to produce an electro-osmotic flow for separation of ions, and then it enters into the mass spectroscopy. So it is very important for you to know about this type of, what is the meaning of hyphenated detection technique. Hyphenation detection technique, you should write very clearly, is a technique developed for coupling of two type of instruments, or separation technique in online respect for two type of, or more than one, or coupling of two instruments, or coupling of two technology in a same analysis. That is why it is called hyphenated detection. In that example, you can see gas chromatography connected with mass spectroscopy, liquid chromatography connected with mass spectroscopy, liquid chromatography connected with the IR spectroscopy, and liquid chromatography which is connected with that is nuclear magnetic resonance spectroscopy, and you can see that is capillary electrophoresis which is connected with mass spectroscopy. So these are some examples which we should mention about the hyphenated detection techniques. In the context of pre-isolation analysis of crude extracts in a herbal drugs, herbal formulations, or fraction of various natural sources, isolation and online detection of natural products can be used for chemotaxonomic study. You should know what is chemotaxonomic study. Taxonomy means you should know that there is very classification of herbals, classification of plant sources, classification of plants or florals, how they are done by means of chemotaxonomy. Chemotaxonomy means you should not, that is zero taxonomy, chemotaxonomy, by means of chemical identification constituents, even you can find out by means of this hyphenated detection techniques will be more useful for you to go for two instruments connected in an online basis, and so that it will be giving very good results to find out the chemotaxonomy studies. And chemical fingerprinting and quality control of herbal products, de-replication of natural products, and metabolomic studies, even various metabolic studies, metabolomic studies can also be performed by means of this hyphenated detection techniques. So if they are asking for a short notes or an essay question or in a 5-mark questions, you should be able to write. If there is short notes, you should write what is hyphenated detection techniques. And even I have taken the 2D type of techniques also in same chromatography. 2D techniques are also there. Two chromatographic techniques can be mixed up to give a very good results also, it is possible. So these type of short notes they may ask. So you should know about these things, and also even an essay question also you should mention these as a very important points to be performed for standardization and of the quality control of herbal formulation and herbal drugs. So two-dimensional 2D chromatography. What do you mean by 2D chromatography? You might have heard about in the paper chromatography. We are just running the solvent in two directions and finding out the exact spot very clearly. Not only that, apart from that, two chromatographies can also be followed, and it can be detected more specifically, more specific, more exactly, more what we can say, more not only specificity, and the identification of the compound can be more feasible and more exactly, and you can give more confirmation can be given by means of this 2D chromatography. So what do you mean by 2D chromatography? Before going to 2D chromatography, first once to understand difference between multiple dimensional fingerprint generation. Multiple dimension. You will be changing the paper chromatography upside down, and you can just running from left side to right and also from bottom to top. Two-dimensional paper chromatography you might have studied in theoretically, and that is one example, first example, but even it can be followed in all other instruments.
or other chromatography is also multi-dimensional fingerprint generation by hyphenated detection techniques and 2D chromatography. Hyphenated detection technique, just now we discussed about 2 instrumental, 2 technological, how we are giving a specific concentration on the conformation of a compound very clearly and very easily, and also making the time of analysis have been reduced by means of this hyphenated detection technique. The multi-dimensional fingerprinting, the hyphenated detection collects the information from the eluted compounds, while in the fingerprint is recorded. In 2-dimensional chromatography, fractionation eluted from first chromatography system are chromatographed on the second system. Again it is second system, eluted one is again separated in the second system, having different separation property, resulting in increasing the peak capacity, and entire separation can be done. And even if it is not possible to separate completely in the first chromatography, the eluted product is done to the next chromatography, you will get a complete detail about conformation, and you will get even more feasible separation and more fractional separation can be obtained with a very good peak capacity in an entire separation can be obtained. And to reveal all characters, or to reveal of all character of complex high definitions, high performance, high definitions, 2D chromatography was proposed. With the development of 2D chromatography technique, a new era of herbal fingerprinting has been clicked nowadays. The new advantage of 2D chromatography over conventional one-dimensional chromatography, single chromatography, it is obtained high peak capacity with a theoretically equal to product of a peak capacity of 2 individual dimensions, in 2 ways, and you are confirming very clearly what a product can be identified and confirmed by means of this 2-dimensional chromatography. That's what the point which you should mention. At the same time you have to read or you have to listen my lecture, and so that it will be easy for you to write in the examination. However, a major limitation is very long time span needed to reach the maximum capacity. Sometimes it takes some time for the 2-dimensional detection with 2 operative system and 2 chromatography. Literature review reveals that 2D chromatography has been used with different techniques such as 2D TLC, 2D HPLC, even 2D HPTLC, 2D gas chromatography, and 2D chromatography combined size exclusion with reverse phase liquid chromatography in HPLC is also done. So you can see that combination of these 2 chromatographies can be also done, and it can be told as 2D chromatography. So you have now got an idea about what is the difference between hyphenated techniques, hyphenated detection, and what do you mean by 2D chromatography. So you should explain them. They both are having similarity, but when you are using 2 chromatography, then you can say 2D chromatography. If you are using 2 instrumental techniques like combined with a chromatography with mass spectroscopy, or combined with chromatography with high performance liquid, with in IR spectroscopy and liquid chromatography connected with even NMR, nuclear magnetic resonance instrumentation, then you can go for hyphenated detection. And also you can say even 2-dimensional chromatographies can also be, even you can say it is also an example of hyphenated, because 2 different techniques which has been done and possibility. But there is a small differences when you are telling you the word which is 2-dimensional chromatography, we cannot use for mass spectroscopy and a liquid chromatography when they are combined to do an analysis. So that's the difference which the students have to be understand. and you can explain. This is about, now we are going to see about what we know about the first itself. I should have explained this about, and also it should be given, but I was just thinking that it also needed. Sometimes they may ask in a short question, or in a short question, 2 mark question, they may ask what is the application of column chromatography. It was the basic thing. So the student should know for some examples also. Column chromatography, CC. Column chromatography is a separation technique in which the substance to be separated are introduced in the top of the column, which is packed. Usually silica gel 60, silica gel 120, which is packed in a column, long tube, glass tube. With in the class also I have shown, and we have not done the experiment, but still we have explained this about column chromatography packed with a solid adsorbents, usually silica. A different mesh, not silica gel G, because it's not necessary for it to stick in the glass. It should be packed in a tube. So the mobile phase is then loaded from the top by means of using a reservoir. Usually separating funnel can be kept in the top of a column chromatography and is allowed to flow down slowly, continuously through the column. By means of the gravitational force, it will slowly move through the stationary phase. The component low adsorption is a theory which you might study, and affinity to the secondary phase travel faster. It is low adsorption capacity, they move faster when compared with the greater adsorption affinity towards the stationary phase. They will move, greater affinity will have slower movement, lower affinity will have faster movement. That is a simple adsorption technique which we are doing for separation. The component may move faster and move first, whereas that means the component which is greater adsorption affinity will move, the lesser affinity will move faster first, whereas the component moves slowly or eluted out later or last. Applications, when they ask, you should write column chromatography is used to isolate active ingredients. It is very useful in separation compound mixtures. As we might have discussed even in the research also, we should use column chromatography to separate the compound which you have got extracted, crude extract, and we did not had time, and also there was no much support from our, what we can say, from institution side also. They were not giving no time or specific in fact, I don't want to comment on that, but anyhow, it's possible to do, only time should be there. Isolation of active ingredient, it is very helpful in separating compound of mixture. Even I personally, I am very much interested to do, and I have done many times, even in abroad, I have done personally by my own interest I have done, and I got more results and more separations I have done personally. But I don't want to, because there was some comments which was very difficult and it will be very tedious to do, but it is not very tedious. It can be within 2-3 days or 4 days, maximum of a week, we can have some separation by column chromatography. We can identify certain compounds by means of TLC. If a student who have done a project, he can identify a TLC and a spot can be identified a simple extract by dilution and a solvent system. Why can't he can do the column chromatography? Think about it, but anyhow, now at present in this project work, the students were unable to do, thinking that it's very difficult and it takes long time, and there was some discouraging of some of the discouraging talks and gossiping made the student not to go into it. Maybe they may not trust. I'm not telling that I know everything, but still I know something. I have done something. I'm telling the truth. It's possible for me to do, but still I don't know. Anyhow, it's not matter to go for that type of talk. I will just go on because of I got little hurt when they stopped that column chromatography. Anyhow, determination, and it was a very good, interesting for separation of the compounds. Very interesting. Even a burette can be done. Even the column I ordered, and it was in the lab. It was in our store. Column is available. It is brought. after giving an order of the size and everything. TLC also, you may be knowing, the students may be knowing how far the TLC have been utilized, and now it is how it become familiar for all the students, even for the project and the lab, and for the teachers, and also for our labs also. They were very familiar about these chromatography techniques nowadays. And column chromatography has wider choice of mobile phase, automation is possible. Column chromatography is most preferred and the only separation technique in phytochemistry for the separation of active constituents from the plant. See how it has importance. How it has importance. In phytochemistry, if you want to do separation, you have to do column chromatography before going to TLC or any identification. It is a must for any student have to be done by column chromatography. I don't know why it was not preferred for the project, but it is necessary for anyone to go for column chromatography. It's a simple technique. It's a simple technique when compared, and also it is very interesting technique. And it's an initial technique which has to be done for you when you want to go for any type of project or research. And it is a technique which you should not omitted or neglected or ignored for your research purpose. That is my very strongly I used to say, and it was totally by means of some ego or some of the issues made them to stop that column chromatography in their research. So anyhow, the students have to be remember about what is the importance of this column chromatography in phytochemistry and separation of active constituents from the plant. Used to isolate metabolites, biological fields also, and it is used for removing impurities. It's not only for phytochemical investigation, it is for almost all other formulations, all other constituents, even biotechnology and biopharmaceutics. Whatever you are doing for even biological fluids can also be separated. Removing impurities can be used by means of column chromatography. Since the flow of mobile phase is under the influence of gravitational force, so special mechanical devices are needed to carry out the operation. Now we are coming to some of the application of gel permeation chromatography. Gel permeation chromatography, gels, you have to remember gels are used and it acts as a molecular sieve. Size of the particles can be according to the larger size which cannot enter into the pores directly, they will be elute, and the smaller particles and the small, we are determining, we are separating DNA, RNA, these all protein molecules can be determined, can be separated by means of gel permeation chromatography. So gel permeation chromatography is also called as gel filtration or size exclusion chromatography. You should remember gel filtration, or it can be called as size exclusion chromatography. It is a separation technique in which the separation is based on the analyte molecular size, analyte the sample, or the molecular size of the protein, or the molecular size of the DNA, molecular size of the RNA, molecular size of the compound which you are selected, and this is the analyte molecule size plays a major role since the gel behaves as like a molecular sieve. In gel permeation chromatography, the stationary phase is a porous matrix. Porous matrix. You might have studied about this porous matrix even in your sustained release tablets, sustained release study, and various matrix tablets, matrix system. You might have studied about the matrix porous system of a gel made up of the compound like cross-linked. These all can be used as a stationary phase. There is polystyrene, dextron, polyacrylamide gels, agarose gels, etc. Agarose gels we have done, that is a DNA fingerprinting technology which I have given a video and explained in you last 3-4 class before, which I was telling about the HPTLC and DNA fingerprinting. I showed some video. In that also we were using agarose gels for separating the DNA molecules and to identify and to see the combination, see the identification by means of derivatization, explains how we have used that electrophoresis technique which is used in the gel permeation chromatography. That is an example for you at gel permeation chromatography. So you should know about the stationary phase and what is its application in our natural sources, and also what is the application of the gel permeation chromatography. Once you have an idea, students can do miracles in your answer papers. The molecules and the sample are pumped through the specialized column containing, or a sheet containing, such microporous packing material gel which we have shown, like polystyrene, polystyrene, dextran, or polyacrylamide gels, polyacrylamide gels and agarose gels can also be used. The flow of the mobile phase caused large molecules to pass through the column unhindered without penetrating into the gel matrix, whereas small molecules will be retarded. They will enter into the pores, they will act as a molecular sieve, they will enter into the pores, they will enter into the matrix, whereas smaller molecules will be retarded or retaining, and slowly molecules will be separated, and you will get very good separation of higher molecules to smaller molecules can be easily separated. Their penetration on the gel according to the penetration on the gel. The picture shows just a gel which can be seen in the gel permeation chromatography. An application, as we have discussed, the gel permeation chromatography technique is used for separation of proteins, polysaccharides, enzymes, that is synthetic polymers. So you just imagine how it has been used. The chromatography is applicable not only for this herbal drug technology, even when you are doing something, novel drug delivery system or whatever the delivery system which you are using in the pharmaceutical industry or in the formulations, you have to go for knowing about the application of the gel permeation chromatography. Purification can be done for any compounds and can be used to determine the quaternary structure of the purified protein. A protein having quaternary structures, 2-dimensional structure, 3-dimensional structure, normal structure of a protein with peptide linkage can also be identified very clearly by gel permeation chromatography by using enzyme, and they can be separated. How you remember about the DNA fingerprinting video and the explanation which we have detailedly discussed in the previous classes. Separation of sugars can also be done here. Separation of proteins, separation of peptides, separation of rubber and other bases in their use in the size, by means of size they can be separated. So that's why we are using, you are telling that gel permeation chromatography can be also called as molecular sieve or molecular filtration, gel filtration or molecular sieve technology. It can also be used in another word. There is no sample loss. Only small amount of mobile phase is required. So there is no sample loss. Only a small amount of mobile phase is required. Now we are coming to explain about the supercritical fluid chromatography, SFC. But liquid carbon dioxide, when you are using extraction we have studied, but the chromatography is also having more application of supercritical liquid chromatography. So this carbon dioxide can be used, is also having a column, and the mobile phase carbon dioxide is supplied through the column in a pressurized form, same like how you performed the HPLC, and a detector also, UV detector has been used, and pressure have to be used, and also you are using dampener, purger, gas, even though if you are passing through the gas of liquid gas of carbon dioxide, the temperature, because it is a critical, supercritical fluid, a liquid in a supercritical point, you may be knowing it will be in the form of gas, and when it comes to the room temperature it will become liquid. In the room temperature it will become liquid, and when you are controlling by means of the pressure and the temperature, it can be a gaseous form. That is the main technique which they are using. While in a gaseous form they are using as a mobile phase, and you can very easily, the solvent can be easily removed after separation. And the compound which it has been it has been eluted or it has separated, easily you can separate from the mobile phase. Solvent system removing is the biggest task for any type of researcher. So here it plays a major role. For example, so supercritical fluid chromatography is a hybrid of gas and the liquid chromatography. Is a hybrid of gas chromatography and the liquid chromatography. Is a hybrid for gas and the liquid chromatography. And the combination combines some of the best feature of each. The supercritical fluid chromatography permits the separation of determination of group of compounds that are not conventionally handled by either gas or liquid chromatography. If suppose compounds are not unable to isolate or unable to separate in the normal gas chromatography or in the HPLC or in ultra HPLC or in hydrophilic interaction liquid chromatography, so we can do it by means of supercritical fluid chromatography. So it is applied for the separation of the variety of materials such as drugs, herbicides, pesticides, natural and the food products can be easily separated by means of this SFC. SFC, supercritical fluid chromatography. Supercritical fluid chromatography technique is especially useful for the separation of series of dimethyl polysiloxane and oligomers. Please write these examples. They are not very much feasibly separated in HPLC or gas chromatography, but only in the supercritical fluid chromatography they are very good result for separation of dimethyl polysiloxane and oligomers. The separation of compound with molecular weights from 400 to 700 daltons can be done by carbon dioxide as a mobile phase. That you know that supercritical fluid chromatography, the major stationary phase is liquid, that is carbon dioxide. Carbon dioxide. The liquid carbon dioxide. Supercritical fluid chromatography. And even we can use polyaromatic hydrocarbons. Polyaromatic hydrocarbons can be easily separated by supercritical fluid chromatography, which were very much difficult and not unable to separate in HPLC and gas chromatography. And now we are just coming to the hyphenated system. Hyphenated systems, hyphenated analysis by means of connecting two instruments or two technologies, which you are getting very short-term identification, specific identification. And this is the example, first one, liquid chromatography connected with nuclear magnetic resonance. So nuclear magnetic resonance means you might have studied more detail in your NMR spectroscopy. Your teacher might have told about the shielding, de-shielding, how a magnetic field applied to an organic compound, they will get excited and they will get magnetic field, and there will be this magnetic field, they will cause shielding and de-shielding, and by means of the electrons which is present in them, and these things can be easily denoted, how many protons, how many nitrogen even nowadays, how many carbon atom even nowadays, they can find out even various other heteroatoms also can be identified specifically by nuclear magnetic resonance spectroscopy. So this nuclear magnetic spectroscopy is connected with a HPLC or a liquid chromatography that was called LC-NMR. Liquid chromatography, nuclear magnetic resonance, LC-NMR, improves the speed of sensitivity of detection and found useful in the areas of pharmacokinetics, toxicity study, drug metabolism and drug discovery process. The combination of chromatographic separation technique with NMR spectroscopy, nuclear magnetic resonance spectroscopy, is one of the most powerful and time-saving method for separation of structural elucidation. When you are doing structural elucidation, the compound is getting separated and immediately it's giving the details about number of hydrogen atom, number of carbon atom, number of nitrogen atom can be detected even by means of the compound detail. So structural elucidation, it helps in do connected this type of interlinked spectroscopy will be helpful for more an unknown compound and mixture, specifically the structural elucidation of light and oxygen sensitivity substances can be detected very easily. The online liquid chromatography NMR techniques allows the online, we are using online means it is not a net internet, online the connected. The compound separated in liquid chromatography directly enters into the NMR in a streamline. That is why we are telling online, in the line. Allows the continuous registration of time changing as they appear in the chromatographic run. Automated data acquisition, the process of liquid chromatography connected with nuclear magnetic resonance, improves speed and sensitivity of the detection. The recent introduction of pulse field gradient technique, high resolution NMR, as well as three-dimensional technique improves application in structural elucidation and molecular weight information. How much it has been improved and increased in the structural elucidation of a compound. This new hyphenated technique, hyphenated. We studied hyphenated 2D chromatography and 3D techniques, three-dimensional techniques also we come across. So you see that two-dimensional chromatography, three-dimensional technique and also hyphenated technique means coupling the techniques of 2-3 instruments that we are doing very more useful in the area of pharmacokinetic study, toxicity study, drug metabolism study and drug discovery process. How much it has been application? Clearly you have to mention these things. And now we are coming to the next hyphenated technique is liquid chromatography connected with mass spectroscopy. We have seen that gas chromatography connected with mass spectroscopy. Now liquid chromatography connected with mass spectroscopy. LCMS. LCMS is also same thing. After separation it is connected with the mass spectroscopy and it is also an instrument. Very mass spectroscopy is little bigger box. Liquid chromatography we have seen already very sophisticated. Not only this one, very small one also we have seen in our lab. And this is the information about application about liquid chromatography connected with mass spectroscopy. This hyphenated technique becomes method of choice of many stages of drug developments. Recent advanced technique includes electrospray, thermospray, ion spray, ionization technique. So you see, electrospray, thermospray and ion spray ionization techniques. Because the recent years, the compound which is getting separated from liquid chromatography, that means HPLC, which directly enters into the mass spectroscopy, where it should be combusted, it should be ionized. Sometimes we are using that very clearly, that means, for example, ionization by means of coils, heating, combusting, which is also possible. Flame ionizing detector also works in the same manner how the ionization takes place. So flame ionizing techniques can be done by electrospray, thermospray, ion spray ionization technique after HPLC can be connected, liquid chromatography connected with mass spectroscopy, which offer unique advantages of high detection sensitivity and specificity. The liquid secondary ions to the mass spectroscopy. Laser mass spectroscopy with 600 megahertz offers accurate determination of molecular weight of proteins and peptides. Proteins and peptides, very accurate separation in liquid chromatography, HPLC, and directly entered into the mass spectroscopy by means of using 600 megahertz laser mass spectroscopy. This is the technique you have to write example for. The combined techniques. Combined techniques, don't forget. An isotope's pattern can be detected by these techniques. And this is about the end of the class of this various techniques which is applicable. Hyphenated techniques. Hyphenated technique, you don't forget about hyphenated techniques and how we are doing with two instruments, and also don't forget about 2D, and even I have explained about the 3D dimension also. Finally we are told, suppose you are adding with 2-dimensional chromatography along with the mass spectroscopy. So you should know that how importance of all these chromatographic techniques. And also I have incorporated about the column chromatography. It is very, very important for any project work especially. But here in our institution we did not do. I don't know why. It's a simple technique. Even the column is available. It's a simple technique. That's why even it has been added in the laboratory system also, practical session also, they are told to add as a column chromatography. It can be made, the time can be consumed by means of amount and what type of solvent system which you are using, isocratic or gradient system. The gradient system which you are using manually can be reduced, that's all. That's a very simple manner you can isolate. Anyhow, I have given my maximum to give you some more knowledge which I have. There are many things to study. I agree with that is I have very, very minimum knowledge which I am just, that is I am not a latest freshly finished course finished person, or I am not a freshly researcher which who come out. No, I am very, very old and previous, and I am reading like you only. I just see the journals and referring, and I am just transferring my knowledge. But the students have to go in detail for your future and refer more and more. Thank you very much for being with me. I'm not going to take more time. I had a very good time with being a teacher with you, and also you are my second family, and best wishes for your bright future. I'm not going to conclude and stop my lecture. Still more. There is two chapters are there. I will find maximum to finish as early as possible. Thank you. Thank you.